So our project is on protein structure analysis using reverse phase HPLC. I'm Maddie.
I'm Jessica.
I'm Hannah.
So our objectives were to develop a method in which reverse phase HPLC is used to analyze the retention time of proteins in their native, denatured, and reduced forms, to develop an experimental procedure to be published and incorporated into student biochem labs. HPLC is high performance liquid chromatography. It's a form of column chromatography. It utilizes a phase, so solvents, and a mobile phase and a stationary phase. The mobile phase are solvents, and the stationary phase is our column. Separation is based on affinity for stationary phase, different columns for different compounds. So for ours, reverse phase uses columns with hydrocarbon chains to attract nonpolar hydrophobic molecules. So this is kind of what a basic setup looks like. We have our solvent, which is the mobile phase. It is pumped through our machine. A sample is injected in with the solvents. That is pushed through the column where the protein will stick to the column. And then it'll eventually will flow past the detector after a certain number of minutes and goes into the waste. This is what our setup looks like in the lab. You can see the solvents on top, the waste over to the left. Our injector is in this level. This is our column, and this is our computer where we see all of our peaks. So we are looking at how long in minutes the samples remain in the column before eluting, so before leaving. So we see a peak at a certain number of minutes, and that shows how long the protein stuck in the column. So for each form, it stays in for a different time. Chromatograms were evaluated on the computer software at a wavelength of 280 nanometers.
So protein structure is a very important part of our project as a whole. On the next slide, you'll see the building blocks of proteins are called amino acids. They are formed in a chain of polypeptides, and there are about 20 types of amino acids, and these acids determine the protein shape and function. This shows the structure of an amino acid broken up into the amino group and carboxyl group with a side chain. Proteins go through many different structures as they are forming to become a protein. The first is called the primary structure, where amino acids are linked together by peptide bonds in a chain. The secondary structure involves interactions with hydrogen bonds between the carbonyl O of the one amino acid and the amino H of another. The most common types are the alpha helix and the beta pleated sheets. The third structure is called the tertiary structure. It is known as the native conformation. It is a 3D shape that has various interactions between amino acids, R groups, and other molecules. All of these bonds are non-covalent and they are hydrophobic interactions. Disulfide bonds are part of this third or tertiary structure. They are covalent bonds between sulfur residue cysteine R groups, and they are very strong, stronger than all other tertiary interactions. These disulfide bonds are where the reducing agent breaks apart the bonds in our experiment. So throughout our experiment, we observed 3 different stages of proteins: native, denatured, and reduced. The native protein is in its tertiary stage, and it is also a monomer. All of its bonds, including peptide, hydrogen, and disulfide bonds, are intact. Inside of the structure are hydrophobic and nonpolar regions. These proteins should have a short retention time and not have a high affinity for the column. Denatured protein is the disruption of the tertiary structure and the secondary structure to cause the protein to go back to its original primary structure of an amino acid chain. The amino acid sequence is still intact, and so are the disulfide bonds. The cause of this denaturing includes heat shock, extreme pH, and urea. This denaturing exposes the hydrophobic regions of the protein, which gives it a longer retention time and a greater column affinity. The final protein we looked at is the reduced protein structure that disrupts the disulfide bonds, but the primary structure remains intact. The cause of this is DTT and 2-mercaptoethanol. This reduces and fully unfolds the protein, so there is full exposure of the inside regions, and it has the greatest affinity for the column in the greatest retention time. DTT is the reducing agent we use to get the protein to its reduced form. Here's the structural pathway DTT takes to reduce a protein. This slide gives an overall view of the 3 different types of proteins, native, denatured, and reduced. You can see, as I said before, the different qualities of each of these proteins and how they interact with the column. You can also see the structure of the protein as they break their bonds and go from tertiary to primary. The protein that was used to do this experiment was lysozyme from chicken egg white. It is a globular enzyme and it is small and stable. It attacks the cell walls of bacteria to break down carbohydrate chains and destroy the cell. It also has internal disulfide bonds and is present to protect the nutrients from the developing egg of a chick. In this slide, you will see the disulfide bonds of the lysozyme structure and how they separate to expose the inside of the protein to have the strongest affinity for the column in its reduced state. This semester, we moved on to a different protein to see how it would result in our experiment. We chose bovine pancreatic trypsin, which is a monomer globular protein, and it is commonly found in our small intestine to help digest protein. It has 6 disulfide bonds. In the final slide, you will see the structure of trypsin in its sequencing.
Lysozyme procedure. Lysozyme was performed with both DTT and mercaptoethanol. Experimental outline for the native. There was a citrate buffer with a pH of 5.2, 1 milligram per milliliter protein concentration, and the injection volume was 20 microliters. The mobile phases, which were solvents, were water with 0.1% TFA and acetonitrile with 0.1% TFA. Experimental outline for the denatured. The same buffers, solvents, and the same method. Combined with 1 milliliter of 8 molar urea with 1 milliliter of 2 milligrams per milliliter lysozyme in the vial. The final concentrations was 4 molar urea and 1 milligram per milliliter lysozyme. Experimental outline for the reduced. It had the same buffers, solvents, and method. Combined with 1 milliliter of solution with 1 milliliter of 2 milligrams per milliliter lysozyme in the vial. The final concentration was 4 molar urea, 0.1 molar DTT, and 1 milligram per milliliter lysozyme. Trypsin procedure. The experimental outline for the native had a HEPES buffer with a pH of 7.82, had 4 milligrams per milliliter protein concentration, and an injection volume of 20 microliters. The mobile phases were water and 0.1% TFA, acetonitrile, and 0.1% TFA. For the denatured, the experimental outline had the same buffers, solvents, and method. It had 0.5 milliliters of 8 molar urea with 1 milliliter of 8 milligrams per milliliter trypsin in the vial. The final concentration was 4 molar urea and 4 milligrams per milliliter trypsin.
It should say 0.5 milliliters of 8 mg per ml trypsin.
Experimental outline for the reduced had the same buffers, solvents, and method. It was combined with 0.5 milliliters of solution with 0.5 milliliters of 8 milligrams per milliliter trypsin in the vial. The final concentrations had 4 molar urea, 0.1 molar DTT, and 4 milligrams per milliliter trypsin. Wait, is this me?
Mm-hmm. I can do it. So this is our method for all of our proteins. So you can sort of see the amount of solvents, the concentration over time. So for 5 minutes, there's 38% acetonitrile. The purpose of this is to equilibrate and allow elution of buffer and other uranium substances. For 18 minutes, it ramps from 40 to 60%. This increases acetonitrile concentration, displaces protein from the column, and causes elution. For 5 minutes, the acetonitrile is at 100%. This is to remove anything that's left in the column. And then it is brought down to 38% for 5 minutes to equilibrate everything. Go ahead, Jess.
Experimental results. Lysozyme procedure overview. The blank had 1 milliliter of citrate buffer with 1 milliliter of 8 molar urea and 0.2 molar DTT solution. The native had 1 milligram per milliliter lysozyme in citrate buffer. The denatured had 0.5 milliliters of 2 milligrams per milliliter lysozyme in buffer with 0.5 milliliters of 8 molar urea. It was incubated at room temperature for 35 minutes before injection. Reduced with heat. Had 0.5 milliliters of 2 milligrams per milliliter lysozyme in buffer with 0.5 milliliters of 8 molar urea and 0.2 milliliters DTT solution. The sample was heated at 55 degrees Celsius for around 60 minutes before injection. This is the blank. This is the denatured. This is the native. This is the reduced. Lysozyme with 2 mercapta ethanol procedure overview. The blank had 1 milliliter of citrate buffer with 1 milliliter of 8 molar urea plus 0.2 molar 2 mercapta ethanol solution. The native had 1 milligram per milliliter lysozyme in citrate buffer. The denatured had 0.5 milliliter of 2 milligrams per milliliter lysozyme in buffer with 0.5 milliliter of 8 molar urea. It was incubated at room temperature for 35 minutes before injection. Reduced with heat. Had 0.5 milliliter of 2 milligram per milliliter lysozyme in buffer with 0.5 milliliter of 8 molar urea plus 0.2 molar 2 mercapta ethanol solution. The sample was heated at 55 degrees Celsius for around 60 minutes before injection. This is the blank, the native, the denatured, and the reduced.
So our trypsin procedure overview. The blank was made up of 0.5 milliliters of heat buffer with 0.5 milliliters of 8 molar urea plus 0.2 molar DTT solution. The native had 4 milligrams per milliliter trypsin and heaps buffer. The denatured had 0.5 milliliters of 8 milligrams per milliliter trypsin and buffer with 0.5 milliliters of 8 molar urea. This was incubated at room temp for approximately 30 minutes before injection. The reduced was 0.5 milliliters of 8 mg per mil lysozyme and buffer with 0.5 milliliters of 8 molar urea plus 0.2 mol DTT. And this should say that it was injected right after being made. We originally thought it was needed to be incubated for a longer period of time, but we found that was otherwise. So this is our blank. The blank just shows that all further peaks we'll get are from the protein and not from our solvents or buffers or anything. This is the native. The native shows a peak at 10.958 minutes. The denatured shows a peak at 11.086 minutes. And the reduced showed a peak at 11.167 minutes. So some conclusions. We were able to get reproducible results with lysozyme using both DTT and 2 mercapto ethanol. We were also able to get reproducible results with trypsin using DTT. We also learned that the denatured sample for lysozyme must be given the appropriate amount of time to work, and the reduced samples need to be heated for time constraints with lysozyme. That was something we struggled with last semester, but we're able to fix this semester. And with trypsin, it needs a higher protein concentration for peaks to show up. At the beginning, we were using a 4 milligram per mil, or 2 mg per mil concentration with the injections, and our peaks just weren't showing up. So moving forward, we will try trypsin with 2 mercapto ethanol and hopefully get the same results as with DTT. We will write up and develop laboratory experiment protocol and associated student activities for this to be used in a biochem lab. And we will continue to work with other proteins. The next one we will work with is alpha chymotrypsin. The struggle with this one is going to be it splits into 3 pieces when you break the disulfide bonds, so we'll have to figure out how to get that to work. Acknowledgements, we'd like to thank Dr. C, obviously, Jane, Jake, who helped last semester, Emily, who we still use her notebooks to help us now, the Nazareth College Departments of Chemistry and Biochem, all other students who previously worked in this project.